Welcome to a masterclass on the ultimate border control. In this explainer, we're going to dive deep into how your cells manage to separate their incredibly complex internal machinery from the completely chaotic outside world. Every single second of your life depends on this process, so we're jumping straight into the microscopic architecture that literally holds you together. When you look at human cheek cells magnified thousands of times under a microscope, you immediately notice how each cell acts as a distinct, isolated compartment. It's kind of like looking at individual rooms in a massive skyscraper. Today, we're examining the invisible structure that makes this isolation possible. It's a boundary so fundamental that without it, the internal fluid of the cell would simply wash away into the environment. To even realize these boundaries existed, early scientists had to rely on the very first light microscopes. But our understanding today goes far beyond what those early lenses could reveal. Our roadmap for this explainer takes us progressively inward. First, the cell's border control. Second, the cell wall versus the membrane. Third, the fluid mosaic model. Fourth, membrane permeability. And finally, transport mechanisms.
Section 1. The cell's border control, defining the boundary.
Every living cell absolutely must maintain a steady, highly regulated internal state, a concept biologists call homeostasis. And it relies on an incredibly thin membrane to do this. To put the sheer scale into perspective, this membrane measures less than 10 nanometers thick. For context, a single strand of human hair is about 80,000 nanometers wide. Compositionally, we're talking about a layer made of lipoprotein, which is just a scientific way of saying lipids and proteins. Think of it not as a static wall, but as a highly intelligent security checkpoint that actively controls what gets to enter and exit the cell. In some organisms, it even houses the tools and enzymes necessary for cellular respiration and photosynthesis.
Section 2. Cell wall versus membrane, the Great Divide.
A really frequent point of confusion is mixing up the cell membrane with the cell wall. So let's clear that up right now. Think of the cell wall as a thick, rigid brick wall surrounding a medieval castle. It's technically considered dead, it's fully permeable to almost everything, and its main job is just structural support. Plants build theirs out of cellulose. Fungi use chitin, which is actually the exact same tough material found in crab shells. And bacteria use a substance called peptidoglycan. Animal cells do not have a cell wall at all. On the flip side, the cell membrane is like a flexible, highly selective chain-link fence guarded by a top-tier security team. It is a living, incredibly thin structure found in literally all living organisms.
Section 3. The fluid mosaic model, an architectural deep dive. To understand how this flexible, selective fence actually works, we have to talk about a monumental scientific milestone proposed by researchers Jonathan Singer and Garth Nicholson back in 1972. It's called the fluid mosaic model, developed alongside the rise of advanced electron microscopy that really let us visualize cellular structures. This model beautifully describes the membrane. It's not a static, solid shell. Instead, imagine a dynamic, fluid sea of lipid molecules where protein icebergs constantly float, bob, and shift around. It's a brilliant way to visualize how the membrane can be structurally sound, yet incredibly fluid.
Let's break down the actual pieces of this mosaic. The foundation is a phospholipid bilayer. Each of these lipid molecules has a water-loving or hydrophilic head that faces the watery environment on the outside, and a water-hating hydrophobic tail that hides on the inside. You can think of them as introverted fat molecules, teaming up to avoid water at all costs. Peppered throughout this lipid sea are your proteins. Integral proteins are deeply embedded, often spanning the entire width of the membrane to act as structural tunnels. Then you have peripheral proteins, which just hang out superficially on the surface, functioning as communication receptors.
Now the fascinating part is that this entire structure is in constant motion. The lipid molecules can rapidly diffuse laterally, meaning they slide horizontally past their neighbors at a really fast pace, kind of like people shuffling quickly through a crowded room. Occasionally, they can perform a much slower flip-flop move, vertically jumping to the opposite side of the bilayer. But those bulky membrane proteins we just talked about, there's no way they can flip-flop. Their polar regions are simply too massive to squeeze through that dense, hydrophobic inner core of the membrane.
Section 4. Membrane permeability, the selective filter.
Because of this unique structure, the plasma membrane is what we call selectively permeable. This means it acts as an astute biological VIP filter. Because the core of the membrane is made of those water-hating lipid tails, small, non-polar molecules like oxygen can slip right through the membrane completely unbothered. However, the membrane heavily restricts larger molecules, or anything that carries an electrical charge, like sodium or calcium ions. They absolutely cannot pass through the fat layer on their own. They need special VIP access to cross the border.
Section 5. Transport mechanisms, moving across the border.
So how do those restricted molecules actually get in or out? Well, the cell uses a few highly specific transport mechanisms. Simple and facilitated diffusion are what we call passive processes. Imagine rolling a boulder down a hill. It just happens naturally and doesn't require any extra energy. These processes move molecules downhill from an area of high concentration to an area of low concentration for absolutely free. Facilitated diffusion just means a protein channel opens up to help those polar molecules roll down the hill. But active transport is entirely different. It is the only biological process here that actively pushes molecules uphill against their natural flow. Because it's forcing molecules where they naturally don't want to go, it demands a direct, heavy payment of cellular energy in the form of ATP.
This brings us to the carrier concept. Think of a carrier protein as a specialized, microscopic shuttle vehicle parked right in the membrane. It binds its specific target molecule on the outside of the cell. The moment it binds, the protein actually undergoes a physical shape change, a structural modification that allows it to safely carry the cargo across the barrier. Once it reaches the other side, it releases the passenger inside the cell and then simply resets its shape to pick up the very next molecule. It operates a lot like a highly efficient revolving door.
A prime, everyday example of this active transport in action is a sodium-potassium pump. Right now, all throughout your nervous system, this pump is constantly burning ATP energy to force 3 sodium ions out of the cell while bringing 2 potassium ions in. Here's a mind-blowing fact. A massive portion of all the energy your body produces every single day goes directly into powering these specific pumps. This unequal exchange of ions maintains a resting electrical potential of negative 70 millivolts. It's this exact electrical charge that allows your nerve impulses to fire. So quite literally, this microscopic pump is the reason you are able to think, feel, and listen to this explainer right now.
As we wrap up our masterclass, I want to leave you with the fascinating physiological puzzle to chew on. If this cellular border control is so exquisitely selective and these carrier proteins act as such perfect locks, how do invasive entities like certain deadly viruses or bacteria manage to hijack these exact transport mechanisms to breach the cell's defenses? Think about how a flawless security system might just be tricked by the ultimate counterfeit key. It's an incredible microscopic arms race. Thank you so much for joining me in this explainer, and keep questioning the microscopic world around you.